in February, I took a trip to California, and on the West Coast airports in California, I saw people wearing masks, and I said, this has to be serious. I've never seen this before. And that's when I just started saying, okay, let's figure out what's going on. I'm going to start at the very beginning. I'm going to look at the papers that discovered the virus and, take it wherever it leads me. And I never expected to find that those papers didn't actually show any virus. I was really surprised that the experimental techniques that they were using, it couldn't possibly show a new organism. It was totally the wrong experiment to do. At first I was just confused. I'm like, I must be missing something, because why would it be so far off? It was just shocking, and I had to really talk to other people and look at their work and look at the historical context before I could fully appreciate what was going on.
With any virus, to show that it exists, you need to separate it entirely from any biological material.
When somebody says, a virologist says, we have isolated a virus, I thought, as does everybody, that they took, 100 people or 500 people who are sick in the same way, right? They have a runny nose or a cough or pneumonia, and then you do steps, like you take their secretions, you then macerate it, and then you filter it, and then you centrifuge it, and then you have a band that has just those viruses, right? That's what everybody thinks they did. And then you tell people the steps, so some other lab could repeat the steps. That's how you do it in science. And then everybody says, yes, this thing that we have is pure SARS-CoV-2 coronavirus. That's what every human being understands by the word isolation, and that's what every doctor and human being thinks happened in this case, right? Except it didn't happen once.
So essentially what I found was that they were taking the liquid from patients, and they were maybe centrifuging them or filtering them, but I made a calculation that in the 15 micrograms that they were using to do the test, they were still like about 30 billion particles, virus-like particles, of which 85 to 95% of human origin, human genetic origin, and they would take this very heterogeneous material and put it into a culture, and then they would say that that was the isolated virus, which of course didn't make any sense.
They're taking samples from the patient to try to find a virus, but they don't look in that fluid from the patient for the virus. I'm like, well, why not? That's the source of it. Instead, I see they mix it in this foreign cell culture, monkey cells. I'm like, why would they do that? It doesn't make any sense at all. They basically combined the words virus and isolation together and created a new definition for that term. And this is why it's so confusing. So only the scientists or doctors who actually take the time to read the paper and read the methods are going to understand that there's no isolation going on. Everyone else who just reads the title of the paper, they'll just, oh, well, yeah, they said it right here. They said isolated.
They don't actually isolate viruses. They just say that they isolate viruses. If you think of something like a bacterial species, and it is possible to isolate bacteria, as in just get the bacterial cells, separate them out from everything else, and then analyze them work out what their composition is, their genetics, their structure, work out what the bacteria, how they behave, what they can do, etc. And I think most people think that with viruses, they're doing the same thing, except that with viruses, when they use the word isolation, it doesn't mean that they've physically separated and purified the particles. It means something totally different. So that's the first deception.
So the question is, why don't they do it? Why don't they show us this virus from a sick person? Not only in 100 people, but in one person. I mean, so I don't know the answer to that, but what they say is, well, there's not enough of these virus particles in a sick person to find them. Which is odd, because if there's not enough viruses in a sick person to find them, what's the theory that they're going to make us all sick and kill us all? So that's obviously ridiculous.
In the virology papers that form the foundation of the pandemic, indeed, the term isolation is used in an entirely different way from its ordinary usage. What the papers mean by the term isolation is that the nasal or throat swab has been isolated from the patient, not that the virus itself has been isolated from the nasal swab. Without isolating the hypothetical virus, these doctors and scientists argue that no independent variable can be properly studied or characterized. It is the first step in what is considered the bedrock of both the scientific method and the founding principles of microbiology, known as Koch's postulates. Through FOIA requests, an activist named Christine Massey has received confirmation from more than 150 government institutions, including the CDC and NIH, that they have no record of a virus named SARS-CoV-2 ever having been isolated directly from a patient. Instead, these institutions point to scientific papers which use three methods for claiming they have discovered the existence of the virus without ever having isolated a virus itself. The first of these is known as the cytopathic effect.
People have sent me in this past year, let me just take a guess, 50 papers called The Isolation of SARS-CoV-2. Now, maybe 48, but, it's a lot. And both the normal virology community and the sort of alternative community sends me paper after paper saying, here's how they isolated, here's a paper showing the isolation of this virus. Every single one did it the exact same way. They take snot and they inoculate it onto tissue culture, usually monkey kidney cells, which are called vero cells.
And put it in this recipe of the toxic cell culture with, toxic antibiotics, and they usually use ones that are specifically toxic to the type of cells in the culture. So when they use kidney cells, they use antibiotics like streptomycin, gentamicin, and amphotericin B, which are all specifically toxic to the kidneys. They starve the culture, and then they add other biological materials like bovine calf serum, like a fetal bovine serum, rather, like from calves in the womb, their blood, and they can show these what they call cytopathic effects, which is damage to the cells from this every single time. And then when cells are damaged, they make particles like exosomes and apoptotic bodies and other types of particles that don't even have names. And they can just point to whatever particle they want and say, there's the virus. So it's very convenient. It just doesn't come close to proving what they say it does.
The way that they prove, inverted commas, SARS-CoV-2 exists is through stressing cells in cell cultures, vero cell cultures. They stress them with antibiotics, all sorts of mechanisms, and then they see things bud, or they see cilia on the cells stop beating. And so they interpret those as virally caused, or as viruses budding, or as the cilia on the cells, the human cells, they stop beating, so the cell must have died. But it could be due to all sorts of things. very, very uncontrolled experiments, one-shot experiments.
The cytopathic effects are just non-specific effects that occur in a test tube when you put various cell cultures, and then they usually start with a cell culture line, which is highly abnormal. So you don't start with a healthy cell line because they find that they don't seem to do very much in the test tube. So typically they take something weird like a Vero, which is a monkey kidney cell, and it's an abnormal one, so it's got chromosomal abnormalities. And then you use that kind of cell for your experiment because these cell lines are highly reactive, meaning that they have a high propensity to die and break up in a test tube and produce various exosomes and things that the virologists are looking for. So in this regard, they say that all of those effects are due to the presence of a disease-causing virus. But the problem with that is that you can get the same effects by putting other things in the test tube with the cell culture. So you don't need to put a virus in there. It's just the process itself which stresses the cells and causes them to break down. So that's what John and I call the double deception. Firstly, saying that they've isolated a virus when they haven't. And secondly, attributing effects that we can see anyway without the presence of a virus, but saying that it's specifically to a virus. And this is the basis of modern virology, basically. And it's simply fraudulent. It simply doesn't establish A, that there's a virus, and B, that it's able to cause disease.
So here's an article that was in Viruses May 2020. They're talking about now they have a better name for just debris coming from the breakdown of our tissues. They call it extracellular vesicles and exosomes. But that means when we get poisoned or sick, then we break down into these particles. And the question of this article was, can we tell the difference between this breakdown versus something coming from outside, right? So he says, quote, however, to date, a reliable method that can guarantee a separation from extracellular vesicles and exosomes from viruses, quote, does not exist. We can't tell the difference. And essentially what that means is all of these genetic pieces that we're calling viruses are just the breaking down of our own tissue.